So one of the things you learn when you study computational biology is that biological data is inherently very messy. And so you often have two steps. You can try to solve a problem in a perfect case, and then the second part of that always is adapting it to the data that you have. So we've been able to solve the problem of genome assembly if we make some assumptions and we solve it in kind of a perfect case. Now, in practice, we're going to have some complications that we have to deal with. And I had mentioned those when we started because we made some simplifying assumptions, and that's absolutely fine because it helped us solve the problem in this simplified world. But we should probably come back and at least mention the things that researchers do to get around the practical complications in practice. So what I mentioned was, for example, DNA is double-stranded and is broken across multiple chromosomes. So you're never going to, the multiple chromosomes issue means that if we're sequencing your genome split across multiple chromosomes, you're never going to have one contiguous sequence that you're constructing because it's inherently split across different chromosomes. We also saw that the reads may not have this perfect coverage where they, for any region of the genome, you're going to have many, many reads covering it from your data set. There may be some areas that you're missing. And then third, the machines themselves have gotten better, but they're still prone to errors. And they're not going to tell you when they make an error, they're just going to make an error. So let's look at this first complication, which is that the DNA is double-stranded, and so the strands run in opposite directions. So if you're given a read, you don't know, did it come from strand 1 or strand 2? So we're going to need to search because we don't know the genome in advance. So we're going to somehow need to search for it and its reverse complement against the genome. So we need to take both pieces of information, both the read and whatever that read would be on the opposite strand into account. If we're trying to construct one strand, which essentially is going to double the amount of data that we have and mean that half of that data is going to be one strand and the other half is going to correspond to the other strand. And we've got to figure out kind of how to divide this data into two pieces. And that makes things really complicated because we don't necessarily know which strand they came from in advance. We're not going to be able to get this information. So that's one issue. Another is that in practice, we're only ever going to have confidence over reconstructing the genome for a sufficiently long genome if we sequence the genome in pieces. So we may not know exactly how these pieces interact with each other, and we might need to do additional lab experiments to figure out what these pieces called contigs, how they are ordered with respect to each other along the genome, and to construct what are called scaffolds, which are longer contiguous pieces. And then a third complication in genome sequencing is that there are errors. So if we see errors in reads, then in the network that we construct, where the reads get assigned to the edges, then what we see is what's called a bubble. So you may have a path proceeding nicely along the top, and then an error causes a branching in that path where, say, a T got replaced with a C, and we go along the bottom path. You don't know in advance which of those is right because the machine didn't tell you which one was right. It just gave you both. And so we somehow need to find a way of identifying that the top path is the correct one and that the bottom path is the incorrect one, and to get rid of that bottom path, even though the machine won't tell us in advance which one it is. So that's another really practical complication, and researchers have spent a lot of time and energy in exploring different ways of trying to get around this. And of course, it's the type of thing that has gotten better over time as the research has advanced. So it's still a very active area of research. And I guess I would conclude today by just telling you a little bit about the computational biology program that we have here at CMU. So if you happen to be a prospective high school student, we have a new, as of fall 2017, computational biology undergraduate program that's one of only 2 majors available to undergraduates in the CMU School of Computer Science. So up until now, all undergraduate students were computer science majors, and starting as of fall 2017, there are 2 options, computational biology and computer science. So you can check out our program on our website if you're interested at cbd.cmu.edu. You're more than welcome to look me up and send me an email if you have any questions about our program, because we'd love to get in touch with any prospective students who might be interested as well, or to meet them when they're on campus and so on. And the idea of this program is to provide students with a really strong foundation in computer science, the exact same foundation that a CS major here at CMU would have, or just as rigorous, and then we want our students to have a really strong biological foundation, but then to show them the interplay. So we have an entire core of computational biology courses that show the interplay between the computational side of a problem and the biological side of the problem, and show how they mix. And so we think it's a really exciting major for our prospective students, and I, for one, am really looking forward to interacting with our students. And again, if you have any questions about the program or resources for continued education in computational biology, please feel free to send me an email. My email address is pcampos, on my last name, @cs.cmu.edu. And thank you so much for your time, and I would be happy to be in touch with you. Thank you.